All right, today's presentation is going to be on carbohydrates, fats, and proteins. So, carbohydrates are named carbohydrates because they have carbon and water. If you think of them, they're made out of carbon and then H2O. C6H12O6 is the formula. So, the carbon, and then when you hydrate something, you add water to it. So, that's why they're called carbohydrates. Monosaccharides are going to be single-ringed carbs. Disaccharides are going to be double-ringed carbs, and polysaccharides are going to be many-ringed carbs. We'll get to those in just a second. So here's an example of a monosaccharide. A monosaccharide is going to be the monomer for sugars. This is a single-ring carb, and you can see the carbon and then the H2O at each corner. So, carbon and water. So, glucose is an example of a monosaccharide. Sucrose, table sugar, is an example of a disaccharide. Basically, what you have going on is you have two sugar molecules coming together, and you're going to lose a water molecule. And when these form a bond and the water molecule is lost, when we lose water, we call that dehydration. And we're forming a bond, so it's called dehydration synthesis. If we have a whole bunch of dehydration synthesis going on, then we can bond a bunch of sugar molecules together and we get a polysaccharide. Some examples of polysaccharides are starch. This is primarily found as a storage molecule in plants. Glycogen, which is primarily found as a storage molecule in animals, and cellulose, which is a sugary fiber that makes up the cell wall of plants. This is basically wood. So what are carbohydrates? Basically, they're the fruits and the grains. The grains make up things like bread and pasta and such. Beans, and rooted plants, so like carrots and potatoes and those types of plants would also be loaded with carbs. In our livers, we store carbohydrates as glycogen. This sugar can be released when it's needed by one of a couple different hormones. Glucagon will release small amounts of sugar at a time. And if we have a fight or flight situation and we need lots of sugar all at once, then we can have epinephrine will release lots of sugar all at once, also known as adrenaline, and we'll get lots of sugar in our system to be able to deal with whatever situation we're in. Lipids are going to be used for a few different things. Lipids are used for energy storage, and they make up our cell membranes and insulation and shock absorption and lots of different things. They're made up of a molecule called a triglyceride. Triglycerides are going to be made up of 3 fatty acids and a glycerol molecule, and we'll talk about those in a minute. These are your typical lipids you think of with fats as oils and butter and lard. Coconut oil. That's what we usually think of as fat, but actually there's lots of fat in lots of the food we eat, and fat is a really healthy energy source in our bodies. Meats and cheeses are loaded with fat that are healthy for us, and nuts and fish, certain types of fish, and olives and seeds, different types of seeds, and avocados. All these things are loaded with fat, and our body needs a lot of fat to do all the processes it has to do. And fat actually usually gets a bad rap, but it's actually a pretty healthy molecule. It comes in a couple different forms, saturated and unsaturated, and we'll talk about that in a second. Cholesterol is also a type of fat. Cholesterol is going to be used to make all of our steroid hormones, and it's used for stabilizing cell membranes. So this is a typical triglyceride. So you have It's called tri because there's 3 fatty acids, and this is our glycerol molecule over here. And if you have the fatty acid completely filled with hydrogens, we call that a saturated fatty acid. So it's holding as many possible hydrogens as it can, and they make nice straight chains. So these two would be examples of saturated fatty acids. This one's missing a couple hydrogens because of a double bond between the carbons, and we would call this an unsaturated fatty acid. If it has one double bond, we call that a monounsaturated fatty acid. And if it has more than one double bond, it's a polyunsaturated fatty acid. So here are some examples of saturated fats. These would be saturated, and these would be monounsaturated fatty acids. And then down here we have an example of a polyunsaturated fatty acid. Poly, and then a mono between. We can also make phospholipids out of triglycerides. Basically what you do is one of the fatty acids gets replaced by a phosphate group, and you end up with a molecule that looks like this. It has 2 fatty acids on it and a phosphate group, and this is what makes up our cell membranes. The phosphate group is attracted, it's polar and it's attracted to water, and the fatty acid tails are non-polar and they're not attracted to water. And so what we end up forming in our cells is a bilayer where the fatty acid tails can point to each other and the phosphate heads can point both outside where there's water and inside of the cell where there's water. And so the molecules are all happy. Proteins are our last macromolecule. Proteins' role, they defend us against disease. Antibodies help do that. All of our structures are made of proteins, and proteins also make up enzymes, which are going to speed up reactions in our body. They're going to be made up of amino acids. Okay, so amino acids are the monomer for protein. When 2 amino acids bond together, they form a peptide bond, and we lose water, so dehydration, and we form a bond, so it's synthesis. So we got another case for dehydration synthesis right here. When we have a bunch of amino acids connect together, we form a polypeptide. Okay, so when we're dealing with proteins, we're going to have a few different levels of structure. The primary structure of a protein is the order of the amino acids. The secondary structure of a protein is the alpha helices and beta sheets and linear portions of the protein. The tertiary structure is the 3-dimensional structure, and then if you have to have multiple polypeptides come together to form a protein, we call that quaternary structure. So here's primary structure. It's the actual order of the amino acids in the chain. And then secondary structure, you see sometimes things are linear. Sometimes we form alpha helices, and sometimes we form what we call beta pleated sheets. Okay, those are 3 different kind of secondary structures. And then the tertiary structure is how it all comes together. So we see lots of alpha helices and lots of beta sheets, and then some linear portions in between, and it forms one big mass of order, I guess you'd say, to function at some level inside the cell. Enzymes are going to be pretty important. There's a couple key characteristics for enzymes. They're going to be highly specific for only certain substrates, and they're going to be reusable. And they function best under very specific conditions. So basically you have an active site on an enzyme. The substrate's going to come in, bind. The enzyme will then change its shape slightly and cause a reaction to take place, and you'll get a product. And we will learn a lot more about enzymes later on, but basically their job is to reduce activation energy. They basically get rid of activation energy necessary to run reactions in our body. They're very temperature specific and pH specific, which we'll learn more about when we do our lab, but they have optimal temperatures, so they can function at different temperatures, but there's a best temperature for enzymes. You can see down here the optimal temperature for our enzymes is 37 degrees. But if I'm a bacteria living in a hot spring, my optimal temperature might be a lot hotter. The optimal pH for pepsin, which is a stomach enzyme, is a little under 2, and the optimal pH for trypsin, which is an intestinal enzyme that breaks down proteins, is 8. And so we have a little different scenario. Pepsin won't work in the intestines, and trypsin won't work inside the stomach. As far as energy is concerned, proteins and carbohydrates have about the same amount of energy. Fat has a little over double the amount of energy that protein and carbs do per gram. And the one we never really think about is alcohol. It's never really listed like on a beer how many calories you're getting from the alcohol part of it. It might tell you how many carbs you're getting or how much fat you're getting or whatnot, but it doesn't tell you how many calories you're actually getting from the alcohol. And alcohol actually has a lot of calories in it. There's a reason people get beer bellies. It's because the alcohol has about twice as many calories as carbs per gram. And so there are a lot of calories in a lot of the foods, but then also one we don't think about that much is alcohol has a lot of calories as well. So that's our presentation on the 3 macromolecules. And